Hi, my name is Nancy,
My name is Qinyi.
and today we're going to be talking about epistasis for the Harvard-Princeton Drosophila cross that determines the eye color phenotype of the flies. In this experiment, we used a cross between Harvard and Princeton flies to demonstrate epistasis. Harvard flies have bright red eyes because the Harvard gene is responsible for producing a pigment called xanthomatin. On the other hand, Princeton flies have a more brown color from the pigment drosophila synthesized from the Princeton gene. They each carry a mutation blocking the production of one of the pigments. If both the Harvard and Princeton genes are present, the two pigments will together make up the wild-type red-eyed phenotype.
To set up the first cross, we selected 6 Harvard males and 6 virgin Princeton females. And the second cross is of 6 virgin Harvard females and 6 Princeton males. This was very important because female Drosophila can store sperm from a single mating and use those sperms in the future production of her progeny. So by using virgin females, we guaranteed that the progeny resulted only from our intended cross. And after that, we incubated the flies at 30 degrees Celsius to let them mate, lay eggs, and produce the F1 generation. We used the carbon dioxide pass to put the F1 generation flies to sleep. And under our dissection microscope, we recorded the eye colors we observed and the number of different phenotypes in the F1 generation.
This Punnett square shows the Harvard-Princeton cross. And so we expected 4 different phenotypes. First of which was the wild type phenotype, which has brick red eyes. Second of which was Harvard phenotype, which has bright red eyes. And then the Princeton phenotype, which has brown eyes, and the white phenotype. We expected a 9 wild type to 3 Harvard to 3 Princeton to 1 white uh, phenotypic ratio.
So in the case of Harvard and Princeton genes, when both pigments, drosophila and xanthomatin, are present, they combine to produce the white wild-type red-eye phenotype in Drosophila. This is because the pigments work together to create the characteristic brick red color. However, if one pigment is missing, the flies will show a distinct phenotype depending on which pigment is absent. If only Drosophila is missing, the flies will show the Princeton phenotype with brown eyes. And if xanthomatin is absent, the flies will show the Harvard phenotype with bright red eyes. When both of the pigments are missing, the flies will not have any coloration in their eyes, which is the white eye phenotype. This demonstrates to us the concept of epistasis, a genetic interaction where the effect of one gene mutation depends on the presence or absence of mutations in another gene. The observed phenotypes highlight how Harvard and Princeton mutations can influence one another. The absence of one pigment-producing genes alters the expression of the overall eye color, which shows that these genes do not act independently, but they interact to create the observed traits.
So why is this all important? Epistasis is crucial in genetics because it highlights how genes do not act in isolation but interact to influence phenotypic traits. Understanding epistasis helps unravel the complexities of genetic inheritance, explaining why certain traits may not follow simple Mendelian patterns. The concept is important in studying human diseases and traits as it provides insights to how different genes and their interactions contribute to observable outcomes. In the case of albinism, epistasis plays a significant role. Albinism usually results from a mutation in the TYR gene, which encodes tyrosinase, which is essential for the first step in melanin synthesis. If other genes involved in pigment production are functional, a defective TYR gene can completely mask their effects, leading to the absence of pigmentation. This is an example of recessive epistasis, where one gene overrides the expression of other pigment-related genes, demonstrating how epistasis shapes this condition. That's all. Thank you.
Thank you.